Well, thank you for the kind introduction, Sebastian. As you said, I'm Matthias's first student, and I've laid claim to that title for about 30 years now. I don't know the exact date when we met, but I still remember the experience. So I was doing some bachelor work in Professor Peter Röpsdorf's lab, and I was about to begin my master's work, and I came to the lab one morning, and Peter Röpsdorf told me work with this German guy. I thought I was doing and see him now. So I went into a kid pounding away on a Macintosh computer, and so I said, "Are you going to do your too?" I said, "No, I've done that." "Oh, are you going to do your PhD?" No, I've done that. I was most impressed because he was so young and already way ahead of me. I thought he was about my age. So anyway, we're going to rebuild a plasma desorption mass spectrometer, turn it into a MALDI instrument. And that became quite an interesting project for me. And it's actually also an area where Matthias has made, in fact, a very significant contribution when you think of it. You describe yourself as a one-trick pony, perhaps, but if there ever was such a thing as a multi-trick pony, you would be the finest specimen. I mean, if you look at the number of innovative ideas that have come from Matthias's, I shouldn't say hand, but head, over the years, it's mind-boggling. The list goes on and on and on. But actually, already with the MALDI instrument, he figured that, so you only had very few MALDI instruments at the time, back in the early, it was in 1990. He figured that with Hillenkamp and Karas's group using a YAG laser quadrupled wavelength at 266 nanometers, and the child lab was using it at triple wavelength, so at 355. He figured that a nitrogen laser at 337 would also work. And keep in mind that a YAG laser is like a $200-something thousand very, very costly big instrument, whereas a nitrogen laser is like a sub $10,000 device. And then you keep in mind that there's about 10, 15,000 MALDI instruments out there. Do the math. I mean, he has saved the proteomics community over $2 billion just based on that. It's quite amazing. So anyway, one of the first days in converting this plasma desorption mass spectrometer, we needed to build something like it was a pulse detector for the trigger of the time-of-flight spectrum, and he says, "You do that." And I said, "I'm a chemist. I don't do electronics." "That's okay. I have a book," he says, and then he hands me The Art of Electronics by Horowitz and Hill. It is 1,115 pages. I still remember it must be a joke, but then I reminded, oh, he's German. It's not. This is serious, right? I have to actually read this book. And and in fact, over the years, he has lent me, must be close to 100 books that have taught me a lot. I haven't actually taught him anything in that process because I'm the type who never returns borrowed books, and he still keeps bringing me books. And so anyway, probably one of the very first books he gave me, because he reads everything, every type, business, social sciences, and probably the third or fifth book he gave me was a marketing book. And that actually caught my interest more than perhaps anything. At this day and age, I'm probably more of a marketing guy than I'm a scientist. In fact, a lot more. Now, you probably think of marketing as being people who make broad claims and shout to the world and lie all the time. That's not all parts of marketing. Marketing also involves what we call strategic marketing, which is finding out what is needed, what kind of holes to fill, and then if you want to fill that hole, if you want to get from A to B, what is necessary for an instrument or a service or a device or whatever. That's actually the part of marketing that I like most. It's not the Marcom part where you shout to the world. It's finding out what is really needed. That is to say, I'm not lying all the time. But I do enjoy for one thing, there's less work in marketing than in science, but also we have this flexibility. I do appreciate the exactness and accuracy of science, but I do like the flexibility that we have in marketing of treating things a little bit differently. So anyway, let's get started. I would like to talk about the front end. I was actually hoping that somebody this morning would have shown sort of like the entire workflow in proteomics as it's mostly practiced today. You start out with a sample, you digest it down typically, clean it up, inject into an LC system, goes into a mass spectrometer, you get data out the other end that then gets processed. Nobody has actually shown that. So I'm starting, and I would like to talk about just one piece, which is the front end to the mass spectrometer. How do you actually get stuff into the mass spec? And Matthias suggested actually that I should start with a little bit of history. I said, yeah, sure, that makes sense. I didn't realize that although electrospray is a relatively recent technology, it actually predates both the internet and PowerPoint. So I didn't even have a single slide that would actually show anything about the history of electrospray. So the following few figures are stuff that I've copied from various sources on the internet and quickly stuck into slides. So anyway, as was said before, electrospray as an ionization method to be used in mass spectrometry came out of Fenn's lab in the late 80s, so 88, 89, where Matthias did a tremendous amount of work over there. Probably not all single-handedly, but I like to think that it was a lot. And then roughly around 90, when, for instance, in Odense and other places on the planet, it became an ionization technique that people started to deploy, for instance, to analyzing proteins. Prior to electrospray and MALDI, you could do spectra with fast atom bombardment, but those were usually sitting on a sector instrument, very, very trouble-prone. And there was also plasmid absorption, which is using the fission event of a californium atom in order to ionize ions. That was a very, very slow and not very sensitive mass spec techniques, and probably there existed around 10, 15 commercially built mass spectrometers of that kind. So this was really the starting point of when protein mass spectrometry started going faster. Early days, it was really, really slow moving, and it was not without problems. Of course, that's how everything is in its infancy. Fundamentally, when you do electrospray, you form ions by having an emitter. Is this a pointer? Oh, having an emitter, which would be typically a steel nozzle that was at a raised potential of a couple of thousand volts, positioned across from a grounded metal surface where you have a hole that will lead into a vacuum chamber. And that draws out liquid in droplets, and the solvent would evaporate and leave charges on the surface of the droplet, and in the droplet you'd have your sample substance that you want to analyze. And then whether you actually have all the solvent evaporating and leaving the sample hanging in the air with the charges on, or whether it comes to a point where the droplets become so small that the actual sample gets ejected with charge, that process isn't quite understood yet, even after 30 years, and it's probably also not very interesting for the use of mass electrospray mass spectrometry. But this is how it got started. And you'd basically use a syringe drive to infuse quite slowly your sample of interest at pretty high concentration. And it was a very trouble-prone technique. When you think of it, you're trying to put water into a vacuum system, and almost by definition, that's going to be difficult. And the early designs for mass spectrometers were not with differential pumping that worked as efficiently as they do today. So in these early days, I was actually happy to be more of a MALDI person, because that came off to a much, much better start than electrospray. And in 1992, it wasn't really clear that electrospray would ever become something of use to anybody. Then, after having moved to Heidelberg, Matthias was joined by another Matthias, was Matthias Wilm, and the two of them quickly deduced that you would get much, much greater sensitivity in electrospray if you miniaturize the flow rate. This, fundamentally speaking, the slower the flow, the lower the flow rate, the smaller the droplets and the more charge you would get on the droplets per sample molecule. So the net effect was that you should squeeze down your flow rates as low as possible. And they developed very, very intricate technologies, here using borosilicate glass emitter that was gold palladium coated to make them electrically conductive. And then you take like a microliter of your cleaned up substance and directly infuse into a mass spectrometer. Here you see one of these emitters. It's actually pulled to a very, very fine tip here at the end, which would be only a couple of micrometers in diameter at the opening, sitting in front of a SciEx mass spec orifice. And it was incredibly tricky to actually use. It was a very acquired skill. Not everybody could do it. It was even so bad that you start out pulling the emitter, it was actually closed, and then you'd have to gently rub the emitter against the interface so it would break off the tip. And if you broke off too much, then the entire sample would come squirting out and it would be lost. But if you broke off just enough, you'd actually get a very stable flow. Here you see it, the flow profile from such an experiment where like a microliter gives you like half an hour's worth of running. And what you would then do is you actually sit there manually selecting parent ions for fragmentation. And I see Bernard is smiling because he did that quite a lot. And there were really, really experts like Matthias Mann, Bernard, Andrei Shishchenko, who'd go into the mass spec room and sit there for hours, manually picking out precursors and getting fragment spectra. And the trick was to sit on a precursor just long enough to accumulate a significant signal to noise on your MS spectrum. So that actually became the foundation for my first company called Protana. We actually turned these into kits this is a manipulation stage that allows you to touch the emitter against the orifice. We sold them these types of kits to users all over the world, and that sort of became half of what everybody in the world was doing in this age, say between 94 and roughly 2000. The other half of the world was using chromatography in front of the mass spectrometer. And I know it's roughly half-half. So half of the world was following the Matthias Mann school. The other half was following sort of like Dick Smith, Richard Caprioli, John Yates type of thinking, putting a chromatography system in front of. I know it's half because I was selling the ion sources for both, so it's roughly a split world there. But it also applied actually that in order to get any kind of sensitivity in chromatography, you need for electrospray to be performed at very low flow rates. And the flow rate you need is linear through the column has to be constant. But that means that in order to get to lower flow rate, you also need narrower and narrower columns. And at that point in time, what you could buy commercially for chromatography systems, they would all be high flow. And what people then would do is you actually take your chromatography pump and you'd split the flow in a split rate that would be like 100 to 1 or 1,000 to 1. Unfortunately, that means that if you had any kind of restriction to your flow, like a column clocking up, the split ratio would change dramatically and your peptides would be all over the place. So that was actually a very, very, very trouble-prone approach, the only thing that you could do. And that was what people were doing basically from 95 to 05. And it was so trouble-prone that essentially you needed to have one full-time equivalent person just managing and keeping those systems alive for every like 4 to 5 systems. I mean, it took a full-time person to keep them alive. I'm not talking about feeding them with samples, just keeping them operational. So that actually made the foundation for my next company, which was called Proxion. With my marketing hat on, I thought, so what does this world really need? Oh, we need a split-free system that would actually work. So we got to work designing something which we eventually ended up calling the EasyLC. It was meant to be a lot easier to use than a split-flow system. We intended it to be so easy that you would install it like when you get a printer. You just follow 5, 6 steps on a drawing, and then off you go. It turned out to be not that easy, but what we did do was make pumps that would actually have the flow rate that you intended to use, like 250 nanoliters or so per minute, that would then mix the gradient at high pressure, run it through either a trap column or not, and then you'd run it through the mass spectrometer. They turned out to be a lot easier to use, and I think this is fundamental. This is now the schematic from the ECLC. But fundamentally, all the commercial instruments on the market that are used today are using roughly the same plumbing principle, with variation, but it's roughly the same idea. And they are still being used today. So this box actually was designed around 2002, 2003. We thought this is what the world needs. Apparently other people thought the same, because by the time we actually launched the instrument, there were these other competitors already actively selling. So it should have been a blue ocean strategy, what we call it. It turned out to be red ocean, but eventually we pulled through. We sold the company to Thermo in 2010, and at this point in time, there must be somewhere between 3,000 and 5,000 of these units out there. So none of that, however, is very useful in the clinical setting. So nano LCs are trouble-prone still. They're definitely not used for high throughput use. And in 2016, we decided to start over from scratch, design something that was specifically tailored towards clinical use. And that became the basis for Evosep, my current company. And we had one idea that is actually also a Matthias Mann lab invention. This is use the stage tip. We now call them Evotips. But we use them as basically a disposable trap column. So the idea is that you can load your sample offline using a robot or a centrifuge. You can load multiple tips at a time. And then we modified an autosampler to simply pick up one tip at a time with the sample loaded and purified, hold into a funnel, and then run a gradient through. We found a way that we only elute up to a certain percent organic, which leaves a lot of the junk that you don't want to see in your mass spectrometer, leaves it on every tip that then get discarded after the experiment. And you only get the peptides that you want to see into the mass spec. So this would be your standard workflow, and loads of people around the world in the proteomics community already use these tips for desalting, but then they would elute, dry down, and resuspend the sample, then take the resuspended sample and stick it into an LC. In our workflow, you do the digest, you do the desalting, but then take the entire tip and stick onto the LC. Then, in addition to that, we came up with a totally different flow schematic, or a different diagram of how the plumbing should be. And starting out in 16, we basically sat down for 3 months thinking about, so if we want the instrument to behave like this, we want it to be like this, how should it be plumbed up? And we did various tests, and eventually Nikolai Backe, who's also in the audience here today, came up with this really, really silly scheme. That's at least what I thought in the beginning. It actually turned out to be very, very effective in many ways since then, but at first glance, it looked very silly. So what we do, we have actually 5 pumps, 4 of them being low pressure, 1 being high pressure. So at low pressure, but at high flow rate, we actually we form a gradient through the tip, going from, say, 0 to 35% organic. That pulls out the sample that we want to see, but leaves behind the junk, as I said before. Then on the fly, we pull this gradient to a lower percent organic. So something that came off the tip at, say, 10% organic is somewhere inside the gradient, let's say 5% organic. And then we saw the entire gradient in a loop. I mean, this loop is actually like a 4-meter long piece of fused silica, 100 micron ID. So the entire experiment is in the loop. It's created at low pressure, but high flow rate, so like 30 microliters a minute. And we bring the gradient all the way down to this point. And then we switch the valve and use the single high pressure pump to squeeze the gradient with the sample embedded onto the column. And when every peptide species come to the head of the column, it sees C18 phase again, but it's now again in a point of the gradient where it's a lower organic than where it first eluted. So it binds in a very narrow band to the head of the column until a later point of the gradient when it begins to move faster again. So we get some kind of stacking effect from that. So it's important to note that we can make the gradient, move it very close to the column at high flow rate, and that we only have a single high pressure pump. We boxed this up with standardized methods. Here we're trying to show the orange is the overhead of the experiment, and the green is the actual measurement time. And that is a totally different ratio from a normal nano-LC. Normal nano-LC, you can only get your overhead down to like 15 minutes, basically, 15, 20 minutes. So making a really short gradient on a nano-LC doesn't make sense. If you only have a couple of minutes dilution time, but you have 20 minutes of overhead, it's a very, very poor use of mass spectrometry time. But for us, it actually does make sense to go fast. And we have methods that go anywhere from 30 samples per day up to 300. We put a box, and it really was a box. It was a wooden box here in Matthias's lab in 17, I think. No, it was late 16, early 17, where Philip Geyer put it through its initial tests. And lo and behold, he was actually proving that using these disposable traps, you could actually save the separation column from a lot of junk. And you could do injection after injection. Also a nice thing, and here we show that even after 1,500 injections, you don't have much of a back pressure increase. And also what we noticed was that the carryover, because you're using disposable traps, would be very low from run to run. He repeated the same type of analyses with plasma, so it actually should work in the clinic for plasma analyses. And that is indeed what we've seen by now. So at this time, we have around 60 systems out in the field with different labs, different customers. And we have increasing confirmation that it actually works in plasma analyses and should be useful in the clinic. I brought a few quick examples that I'm just going to quickly breeze through. Here's a data set where it was actually a mass spec demo at Buka, where, again, in the old days, you'd bring 1 or 2 samples. Here he brought 192 samples from actual patient material, looking for sepsis. So the scientific content of this is maybe not that crucial, and whether the identifications are many or not, that's besides the point from my perspective. What I note is these green dots. So these are every 10 samples we injected a pool sample from a multitude of the plasma samples, and we see completely stable performance across the entire set of experiments for 200 injections. There's very, very little variability. So the front end doesn't wear out. It keeps going. So I'm running out of time. I'm just going to quickly go through some more experiments. It'll also have perhaps application in microbiology. Of course, microbiology is an area in which proteomics has already made a very, very big inroad. I was talking to a microbiologist who was running the microbiology lab at the University Hospital in Aarhus. She said that the advent of the multibiotyper is the biggest breakthrough in 30 years in microbiology. So huge benefits to go from doing biochemical tests to actually identify the protein profile with MALDI. However, you still have to do bacterial culturing when you want to identify with MALDI. And we have a collaboration with Thermo in Bremen and Florian from Quebec University Hospital, who has the idea that you can actually do direct identification directly from the sample. And thereby, you actually cut the culturing time, which in many cases mean that you don't have to use broad spectrum antibiotics to treat the patient, which would be a major benefit. And she used as a case study urinary tract infections, which are affecting quite broadly and very, very individuals on a daily basis. And her idea was you actually do a very quick sample preparation, do mass spectrometry, get data-independent files, use machine learning to find the best predictive model to see which peptide pattern actually is most indicative of whether you have this or that infection. And again, from our perspective, the nice part, not that we can, I mean, this is still in its infancy, and we can't really judge how effective or that has been and where it's going to go in the near future. But what we do recognize is that all the samples are actually going through the injection phase without any quarrels whatsoever. It just injection after injection after injection, so the front end actually works. It also should be said that these were digested urine samples that were just digests of urine loaded onto our tip, and that was all the workup there was to it. So it is incredibly robust when it comes to taking biological debris. Here's an example from Here's an example actually where the Evosep sits in front of a triple quadrupole. This data is done by Agile in Santa Clara. And again, one of the first tests they did was to actually run 6 plates of 96 samples, so 574 plasma digests onto the system. These, what you see here, the retention times. There's some shady stuff going on here in the middle, perhaps not even instrument related, but what you do notice that this completely flat retention times and no effect to the column whatsoever after 574 injections. What they also did was, this isn't changing as it should. There we go. What they also did was to run actually serial dilution of isotope labeled peptides both before and after the injection of the 574 samples. What they did see was that they're going all the way from 4 attomole peptides spiked in up to many femtomole, I think. Well, anyway. And across the entire serial dilution, for one thing, the highly linear response. But what you can see before and after the injection of 600 samples almost, you get virtually the same response and the same quantitative level. So again, it seems to us that the front end actually works and is ready to do what it should do in a clinical setting. So I'm going to quickly step to this. So far, our intent was to make a system that works for plasma proteomics and working on body fluids. But then there's also interest in looking at small amounts of cells and tissue coming from pathology biopsies, for instance. And after about a year of nagging from Matthias' side, I nagged my people for about a year. And then we actually eventually came to the point where we made a gradient that ran at a much lower flow rate. So, and it actually turns out that the instrument, although it wasn't really intended to run at low flow rates, it does it really, really well. So when you want to run at low flow rates with a conventional nano-LC, you're forming the gradient here, this place, and you're trying to ramp up the pressure. And normally you say that liquids aren't compressible, but that's not true. If you're ramping up to 1,000 bar, you actually compress the B solvent by 15%. So because the pump itself has a large volume, the compressibility of the B solvent is many times more than the actual volume of the gradient. So what happens when you ramp up the pressure in this type of system is that you actually squeeze the A solvent, which is only compressed by 3%. You actually squeeze the A solvent into the B line. And then when the gradient gets going, initially the B pump runs very, very slowly. Then no B actually comes out into the gradient. And what you eventually see is that your peptide is going to be all over the place. It's not reproducible at all. But for us, it doesn't really make a difference because we're still making the gradient at low pressure, moving it to this point, and then we use a single pump to push it out. So for that reason, it seems the system lends itself really, really well. And here are some data from Matthias' lab just showing how stable a 40 nanoliter per flow rate gradient actually can be. And I think we'll hear more this afternoon. Here are some of the first data to just show that you actually do identify quite a large number of proteins from very, very, very small amount of sample. Our work is still not cut out for us yet. I mean, we're still not done yet. As you can see, the first peptides actually elute after about 40 minutes, and then they come in only 30-minute time window. Of course, we need to fix that method so that we spread it out so they come much earlier, and we actually have a much bigger elution window. So we now have the idea of introducing a whole ecosystem of specialized methods for a variety of different things. It could be single cell analysis, doping analyses, a data set that I skipped, small molecule and microbial identification. Initially, we wanted to just make 5 methods totally boxed up more or less like the black box you would need to have in a clinical lab. Oh. But at this point in time, we think we can make tailor-made methods that would actually be specialized for distinct types of analyses in the clinic. And that then would be much more efficient. I'll skip directly to the conclusion. That is that at this point in time, we think the front-end piece is almost ready for clinical proteomics. It's no longer the weakest link. It was for a number of years, but we actually expected to see it now being used. Right now, it's used on a research basis. It will also be used, we believe, in real-life clinical use to work on patient material and to actually lead to treatment, therapies and monitoring of therapies in the real clinic in the near future. So with that, I should stop. I've gone way over time. So happy to take any questions.